Hi, so today we're going to talk about tissue typing. So everyone has several antigens located on the surface of his or her leukocytes. So remember, a leukocyte is a type of white blood cell, and the antigen is the protein on the surface of the white blood cell. So one particular group of these antigens is called the HLA, or human leukocyte antigens. The HLA is responsible for stimulating the immune response to recognize tissue as self versus non-self. So it recognizes your organs for your organs, but if you were to transplant someone else's organs into your body, it would recognize it as non-self and cause an immune response. So it's controlled by a set of genes located next to each other on a chromosome 6, and this set of genes is called the MHC, or major histocompatibility complex. So if you take a look at human chromosome 6, it's on the short arm, it's located here, and these are the genes. The test that determines which HLA antigens are present is called the tissue typing, or HLA typing. Tissue typing identifies the similarity of the antigens present in both the donor and the recipient. So the closer the HLA antigens on the transplanted organ match the recipient, the more likely the recipient's body will not reject the transplant. So the closer they match, and I'll give you examples of this in a minute, the more likely you won't reject it. If your HLA antigens don't match your donor, you're going to have a problem. So for this reason, tissue typing of the kidney donor and recipient is necessary before any kidney transplant. So there are two main classes of HLA antigens. There's class 1, which includes A, B, and CW, and there's class 2, which is DR, DQ, and DP. Every person inherits each of the following antigens from each parent. So you have two sets. So you get A, B, CW, DR, DQ, and DP. That's from one parent. And then you get another set from your other parent. So you get a total of 12 different HLA antigen genes. So here they are, and here's where they're located in the MHC complex. You have all of the genes here. So the set of HLA antigens received from a parent is called your haplotype, because it's half of what you get from one parent. So remember, you're going to have two haplotypes make up your actual genome. So there are a variety of alleles for each of these HLA antigens. So on top of A, CW, B, DR, DQ, DP, there's also alleles. And those are in terms of, they list those in terms of numbers. So the large number of possible variations and combinations of HLA antigens make finding a match in a family much more likely than finding a match in the general public. So you get 12 HLA genes in total from your parents, and along with that, there's different alleles for those 12 genes. So when performing an HLA typing test for a kidney transplant, we look at only 3 of them. We look at the A, the B, and the DR. The MHC genes are the most polymorphic known, so there's hundreds of known alleles for each HLA antigen. So each allele is identified by a number, like I said a little bit earlier. So there's A1, A2, A3, etc., etc., etc. So there's a lot of different combinations of HLA. So 6 HLA antigens are looked at for each person. Remember, each person has 2 of each of the antigens, one inherited from the mother and one inherited from the father. So you can, if you take a look at the mother's and father's haplotypes, okay, so we got, the mother has A, B, and DR, but she's got allele 9, 4, 2, 3, 6, and 15. The father has 29, 18, 5, 1, 7, 11. Remember, the child is only getting half from dad and half from mom. So this child happened to inherit the 9, 4, and 2 from mom, and the 1, 7, and 11 from dad. By analyzing which 6 of these HLA antigens both the donor and the recipient have, scientists are able to determine the closeness of tissue matching. So a 6 antigen match is the best compatibility between a donor and a recipient. This match occurs 25% of the time between siblings who have the same mother and father. So it's, once again, you're doing probability the probability of getting 2 children with this exact haplotype is only 25%. But you want as close a match as you can get in order to get a successful transplantation. So HLA typing techniques, traditionally it was done using serological techniques. The blood from the patient was mixed with serum containing known antibodies to determine which antigens were present. Now it's predominantly done using molecular techniques, so isolating the DNA, using PCR to amplify the genes, and then the genes are sequenced to determine which alleles are present. The second way is much more accurate. So once the donor and recipient have been tested for tissue compatibility, the next step is antibody screening, also known as a panel reactive antibody or PRA. This is just a small amount of the organ recipient's serum is mixed with the cells from 60 different individuals, and each test is done separately. So the purpose of it is they can determine how many different HLA antibodies a patient has in his or her blood. So if the patient reacts with 30 of the 60 cells, he or she is said to have 50% reactive antibody. The lower a person's PRA, the less likely he or she is to reject a transplant. Then a CrossMatch test is done. So after tissue typing and antibody screening, the final test is CrossMatch. And a small amount of the potential donor's white cells is mixed with a small amount of the recipient's serum. By exposing the donor's HLA to the recipient's serum, scientists can determine if the recipient has antibodies to any of the donor's HLA. So a positive cross-match is a reaction between the donor and recipient sample, and it indicates that the recipient's body will likely reject the implanted kidney, and it indicates the transplant cannot be performed. So this is basically if you get an immune response, if you get a reaction between the HLA and the serum, then we can't do a transplant. Negative cross-match is no reaction between the donor and recipient samples, and this indicates the body will most likely not reject it, and then you can do the transplant. And that is all for today. Thanks for watching.